Well, thank you all for joining. My name is Janine Coleman, and I'm the Executive Director of Origins Council. I am one of the co-principal investigators on this amazing study, Legacy Cannabis Genetics, People and Their Plants, a community-driven study. I'm very pleased to be joined today by Eleanor Kuntz and Nick Batura of LeafWorks, and they're going to be offering their presentation and Q&A regarding the work of LeafWorks on the project around cannabis genetics and herbarium science. And so we have a Q&A function that's open, and so if folks have questions as Nick and Eleanor are presenting, feel free to throw your questions in the Q&A. And then we'll be fielding that after the presentation, roughly 45 minutes to an hour in. Wanted to make sure to have a lot of good time for Q&A from you all. And the webinar is being recorded and will be posted to our website probably early next week. And so for folks that weren't able to join us, they'll be able to tune into the webinar via the recording. So thank you all again for joining us. And with that, happy to pass it to Eleanor and Nick.
Okay. Is that showing up? Yeah, great. All right, well, thank you all for joining us today. I know Eleanor and I are very excited to give this presentation and talk about this project. And without further ado, I'll let Eleanor kick it off.
Okay, well, first, I think it's really important that we introduce ourselves and a little bit about our backgrounds so you can know why we're so excited about this project. So Nick and I met in grad school, and we are both population geneticists. My particular background really focused on the intersection between cultivated plants and their wild and weedy relatives, whether they be progenitors that were the ancestral populations to grow into and become our cultivated plants, and then also weeds and other congeners that like to interact genetically between our cultivated plants and our wild borders. So this work really was deeply impactful in me in understanding how gene flow and how different communities of plants talk to each other across pretty wide geographical boundaries. And the structure of the genetics that arises from this kind of crosstalk can give some really clear information into histories of plants, where they've been and where they're going. And that background really set me up to have a pretty profound interest in cannabis, because it's kind of a extreme example of genetic gene flow and crosstalk with a lot of very specific and nuanced environmental, being human mediated and just geographically mediated ways that we've really driven this plant into an exceptional amount of diversity. And so I really am excited to see where this project goes. And that's where my background is. And Nick has some really interesting background as well, so let him introduce himself in his academic history.
Hi, everyone. I'm Nick Batura. And so my background comes from, I've always been interested in medicinal plants. I've wanted to understand why so much of our medicine comes from plants. And I've always thought about that from the plant perspective. Why is it producing all of these really useful secondary metabolites? So that led me to study passion flower, in particular Passiflora incarnata, in grad school, studying under what genetic and ecological and evolutionary forces does it make a larger diversity of secondary metabolites and larger quantities. So I think about everything from the plant's perspective, and obviously cannabis is incredible plant, and it has a lot of secrets to share with us. And yeah, this whole project is really exciting to better understand this plant in particular, how it's been stewarded in the state of California.
So for this particular project, the Legacy Genetics project, we have some very specific goals that the LeafWorks team is diving into to propel the overall research project. So our specific tasks are documenting legacy California cannabis plant diversity. And this includes understanding the history of the plant and creating herbaria and plant vouchers by which we can document that history and that plant diversity in a way that is very fundamental to botanical science. We're going to work with the community to codify names of established cultivars, strains, varietals. We're going to talk a little bit more about what each of those categories really means and why it's important to have established naming systems for our cultivars and our plants. This includes establishing phenotypic boundaries that are encompassed by those names and breeding history and what we would call evolutionary lineage of those plants to help understand the overall groupings and the history of the plants themselves. We also really want to understand how cannabis genetic diversity is distributed. So in any model, any system that you're looking at, if you want to propel something forward or you want to protect it, the first fundamental thing you have to do is understand how that diversity is distributed to do any of those tasks effectively. And so the way that we generally look at this as population geneticists and looking at genetics, not in an individual or at the gene level, but in a whole population, is to understand how that diversity is distributed within populations and between populations. And so within the context of this project, that really translates into within and between communities and regions.
So first we'll focus on this first section.
So plants are exceptionally diverse. That's like the understatement of this entire talk. And we have entire institutions that are really devoted to understanding this botanical biodiversity and cataloging it such that we can understand how to communicate about these things effectively between individuals. Plants are exceptionally wonderful in their ability to produce a wide variety of forms from a very small founding population of individuals. And so a very good example of this is in our agricultural system. So we've seen the Solanaceae, the tomatoes themselves. When you look at the amount of diversity we have in form, growth traits, flavor profiles, chemistries, it becomes very clear that you can push these different populations in multiple directions and codify them in a way where they are producing the same types of fruits over and over. Another really fantastic example that every one of us who's ever been in any kind of intersection between botany and agriculture has studied brassica. So Brassica olearia is a tiny little weed, and we have managed as humans to really propel this plant into all types of forms that we find very useful. So the same exact species produces cabbage, Brussels sprouts, kohlrabi, kale, broccoli, cauliflower, kohlrabi, all of these different things. I think I said kohlrabi already, but and so you can see a single species can be very different in its form. And that nuance is important when we have language surrounding those plants, because we also know that broccoli and cauliflower are the same species, but sorry, we're having a little bit technical. I don't know what just happened.
Sorry, there's a technical issue that we're running into right here.
Of the internet.
1 second, everyone. I apologize.
I am so in love with this book. You guys did a beautiful job. Thank you.
Oh, Google. I apologize for that, everyone. Yeah.
Yeah, great. Well, let's keep going. So we know that so when we look at something like broccoli, for instance, there's not just one kind of broccoli. We have hundreds of cultivars of broccoli as well, in addition to hundreds of cultivars of cauliflower. So you can see that this naming becomes very, very specific, and there's great power in that. And so cannabis is no different. Cannabis is a single species, and we have an exceptional diversity in every single trait you can possibly imagine. Everything from floral morphology to the chemical profiles that are produced, plant physiology, overall agricultural or agronomic structure, its ability to resist pathogens or be susceptible to pathogens. And of course, the list goes on. And so cannabis is a wonderful example of diversity in action. And so the really important work for this project and with our community partners is to really understand from those individuals that have been stewarding the plant and have the most intimate knowledge of this diversity, how do we codify and parse this amount of diversity into a naming system that we can all be talking about the same thing? So one of the most amazing and fundamental resources that we have as botanists are herbaria. And so what a herbarium is a collection of plant samples that are preserved through the vouchering process. So like you see on the right-hand side of the slide, this pressed plants that's normally dried and mounted on paper. Some plants you can't do that with, so you do cross sections and dry them. But it's a dried, preserved form of the plant that can be referenced for hundreds of years. So when we look at herbaria, you have these resources that don't change over time. So you can really learn a lot about how things have evolved. You can look at where they were distributed. And it's very interesting. Our first formal typified cannabis is from Linnaeus. These pieces of paper are called vouchers. So some just like lingo that we'll use throughout the talk. And the first voucher of a plant is generally speaking made when the plant is quote unquote discovered. Of course, we know plants aren't really, they've always been here. It's just the institution of academic science is discovering them and codifying what that plant is, what its name will be, and how we can communicate with each other about our findings around this particular individual or assembly of plants. And so a voucher that is linked to an original description of a new or novel species, whether that be an insect or a plant or an animal, contains certain types of physical descriptors. And with a plant, it is 100% always the use of a voucher in part of that description. So they're very common practice in other types of biology, in addition to species biology, where we're finding new species or creating new types of cultivars and varietals. And they're foundational in identification and then as resource for maintaining and understanding cultivar definitions. So these pictures down here are really interesting. These are from the Missouri Botanical Garden. And you can see these long rows and rows of cabinets. They're specially designed to house thousands of different vouchers or accessions of plants. And, you know, they last hundreds, they last a really long time, hundreds of years. So the origin of herbaria is kind of interesting. Our oldest known examples of vouchers in herbaria really come from the medicinal plant world, which of course both Nick and I are very proud of, being very deeply connected to our medicinal plants. And vouchers were used by doctors to describe to their students what the medicinal plants look like and where they grew so that they could then go out and forage and create medicines for their patients. And so it was this way of transmitting information to create systems of botanical medicine where we are talking about the same plant so we can get the same clinical results from the medicine that's administered. Most botanical gardens in the world house some form of herbaria in conjunction with the living plant collection that they have. And if you look at this map, all of those little red dots are active herbaria that are still actively holding and collecting various collections. Of course, where the herbaria are located will really drive the types of plants that are held in a particular collection. So when we look at our plant of interest in this arena, cannabis is exceptionally missing from the herbaria that exist across the world. We have very spotty representation. Most of it is from 100 years ago, and there's very little genetic diversity that is accounted for with these collections. Of course, we all know why this is the case. This is a direct result of cannabis prohibition. And this prohibition has not only limited our ability to document these plants, but everything that follows from that, which is research, consistent definitions to be able to talk about these things within and across communities. And within the market, it really leads to a lack of quality control that is pretty rote in all other botanical materials that are traded, whether they be for an agricultural food purpose or a medicinal input, whether it's natural medicine or pharmacological inputs for creation of allopathic medicines that you get as prescriptions. So when LeafWorks was really diving into a lot of these issues, it became very clear that the lack of an herbarium was fundamentally it impeded a lot of the work that needed to be done. And the genetic community that was starting to come and surround cannabis wasn't necessarily versed in the importance of the botanical process. And part of any botanical process, as we've already discussed, really starts with this fundamental documentation of plants through vouchering. And so we realized if we wanted to be able to convey consistency in the data and the work that we were doing for our clients and in the communities we were working, we really needed to start an herbarium so that there was this resource. And so Candor was started as a nonprofit. It's the first fully devoted cannabis herbarium that exists in the world. It was founded in 2016. And this really is a function of holding specimens that individuals who have contributed to Candor can reference for their use. So whenever we work with clients, we require them to make a voucher because it adds weight and gravity to the work they're doing, and it gives them a reference point, a piece of prior art, as many of my lawyer friends like to call it, where there's a stake in the ground that this plant is in existence, and there's some tangible link to ownership. I have a hard time with plants. Stewardship, I think, is much better. I don't think we really own the plants. I think we could argue they maybe own us a little bit, but this relationship, it points to the length and the duration of the relationship such that no one can come in and say, this is ours and now you can't grow this. It's a very tangible way of saying, no, these plants have been in community and they aren't owned by anyone, and or I have something new and unique and different and I've done a lot of work, which we'll get into in a moment, and I have something special that deserves some amount of IP protection because of the breeding that we've done. So it really acts as that resource. So the benefits of herbariums in general, but the Candor herbarium specifically, is really the ability to document ownership, preserving biodiversity and history, cataloging temporal, geographical, phenological, and phenotypic diversity, advancing research, and then, of course, this fundamental issue of defining cultivars. So we're kind of going to go over these one at a time. Documenting ownership. This is kind of a philosophical thing, right? So there's ownership in the sense that the community owns these plants. These plants have been with us. It's preventative, it's proactive. It's saying, no, these plants have been with us and no outside entity can come in and say, no, this is uniquely ours and now you can no longer grow it. There's a history of having these plants within a growing region or within the market itself. Within plants, ownership is kind of a hotly debated intellectual property topic, and knowing what's out there is important regardless of if you want to protect something from being patented and or you've done work to show that you have something unique. You know, you need it for both sides of that argument. If you look at preserving biodiversity and history, we have so much history for so many plants. Not only, like, we know how plants have moved across the globe. We know how their expressions are very different within different eco regions that they're grown in. And a lot of this really comes from herbaria, because you can look at a global overlaying of poppies or sunflower, and you can see, oh, they're always taller in this region, or they're genetically distinct over here. And really, so many questions can be answered by leveraging a resource like an herbaria. One thing that's always of a hot topic, especially now in really climate change and understanding how climates are changing and where things can live and persist, understanding temporal, geographical, phenological, and phenotypic diversity across the geographic gradient becomes an immense benefit and resource to those individuals who are interacting with an herbaria. So for instance, if I am a farmer who has a particular cultivar that I'm known for growing, and I'm doing very well, and I want to expand my operation, it could be very nice to use a resource like this to understand what type of property do I need, what type of property partner do I need, such that I'm going to get the same quality out of my plant if I'm expanding there. And or I have a different scenario where I have 10 things that I want to grow, and I don't know exactly where to grow them. Understanding how each of those plants does in that area can help you make good farming choices. So grow broccoli and you live in Ohio, you're going to grow a very different cultivar of broccoli than you would grow if you're living in Georgia. So it's just really using the herbaria to ask very basic agronomic questions that benefit the farmer and the decision makers within an organization. Like, how are we going to make this profitable? How are we going to have the best success? So it's another way you can leverage the information that's found in the herbaria. And then we have more abstract questions that some of our more science geeky individuals want to dive into, looking at specific genes, or maybe it's even looking at agronomic traits. Advancing research, really, you need a lot of data, and aggregating data across an environment becomes important. And when we do this, we always enter specimens into an herbaria, because it's a way to document that academic research and leverage it in future questions, so that you don't have to do the same work over. And then I think the juiciest and most interesting topic that a herbaria can really help with is cultivar definitions. So genetics are variable, right? Siblings are not the same, but they're from the same family. So genetics has a very good perspective on the taxonomic unit, but it's not what really ever has driven cultivar definitions. Genetics is kind of the icing on the cake of what a cultivar is. So cultivar definitions have always come from the communities that cultivate those plants or breed those animals. They know what the phenotypic boundaries are. They know if height matters to the definition, if flower color matters to that cultivar definition. And so really understanding the important things that you're looking for. So when you say something and you have a name, we're all referencing the same thing. So in order to do that and codify and establish cultivar and strain names, the first and most important thing is to look at phenotypic boundaries. So I get this a lot, like, why do we care about names? And I think the easiest answer for me is if you really like Granny Smith apples, you like that sour, sharp bite when you bite into an apple, and I give you a Fuji apple, you're going to be very disappointed in the apple I give you because it's not what you're expecting. And so I think that's the most straightforward thing about a name is that you have an expectation about what that thing is. And it doesn't mean it has to be genetically identical necessarily, although some things like apples are. An apple is only that type of apple if it is a grafted clone of that apple. So it's very similar to some of our cultivars where it's only that cultivar and if it is a clone of that thing. But it doesn't necessarily have to be genetically identical, but we have a prescriptive understanding about what that thing is based on its phenotypic characteristics. Within this particular community and market, we have a really huge problem with naming, and it goes back again to prohibition and difficulty in markets and lack of leveraging or misusing consumer understandings about what names are, where, I mean, everyone on this call, I think this is kind of a no-brainer statement, but a lot of times you'll have a plant and if it's not selling well, you will just name it something else. And all of a sudden you'll change the dynamic of the market for that particular bag of a commodity. And where I think this has been useful or leveraged, in the short term, in the long term, it's exceptionally detrimental to a market. If you purchase something and you like this thing and it has a certain name, and then you buy that same thing with the same name and you get a vastly different experience, it does not boost return customers. It does not help the overall market really propel itself forward to have something where both the inside community and the outside community have the same expectation about what that thing is. And so then we get to how do we do this? It's huge work and it can only be done with the community's input. So cultivar definition. So what does the word cultivar mean? The ICNCP is the International Code of Nomenclature for Cultivated Plants. So this is an international group that focuses on cultivated plant names, and they publish the cultivated plant code. And so if you look up any apple, any varietal of broccoli, you can go to the ICNCP and look it up and you can read all about what makes that taxonomic unit by that name. And so in general, it's an assemblage of plants that are selected for desirable characteristics that are maintained during propagation. And so this should immediately flag, we're talking about domesticated plants. We're not talking about wild plants, we're really talking about domesticated plants that are grown and produced for a specific purpose, and that purpose is linked to the characteristics that are tied to a name. So in general, a cultivar is defined in this way. And like I said previously, it can be very limited such that only a clone is that thing, or it can be very wide where you can have a wider range of phenotypes and genetic backgrounds that fit in that particular category. So we care about this so we can communicate. And I think a really big reason why the cannabis community itself should care about this and really have their own definitions move forward and codified is because if you want to operate in a market where the thing in the bag matters and the thing in the bag will command a different price based on what's in that bag, you have to have names that have meaning. So if I want to make applesauce out of Gravenstein apples, I might pay more for those Gravenstein apples than I will Fuji's because I want that particular apple in my product. And so a differentiated agricultural product can drive a different price, and there can be different metrics for that. So there are many commodities they're just commodities. It doesn't matter what the cultivar is. I studied rice. This is one of those agricultural commodities. It doesn't really matter what cultivar of basmati rice you have. Basmati is a category. It's just basmati rice. I don't care what cultivar it is. I care how long are the grains, how many of them are broken, are they milky white, are they clear? There are other grading techniques that grade the rice, but it doesn't matter what it is in the bag. And that's the opposite of what I think most of us want in this space, where we want the plant itself to drive some amount of additional value based on what we're growing, where we're growing, what we're growing it for. Also, once you move into plants that are specifically being used for medicinal purposes or special utility, it really matters what plant you're getting. This is especially true in the context that we're only testing a handful of chemicals to make any kind of chemotypic description of the plant. And there are hundreds or thousands potentially of chemicals within those plants that have an effect on the usage. And so if you're only looking at 5 chemicals, you're not really looking at the full picture. And so a name that's attached to a certain genetic can get you to a place where now you can have consistency in what you're using. And then you can start to look at the chemistry as a quality metric, not as a metric of ID. And then, of course, we get to IP considerations. And this is both for the community and individuals or individual organizations. Certain plants are fundamentally important to the community themselves. They've been in community for years, generations even, and or their fundamental breeding stock. And so that would be collective IP that you want to protect so that no one can sort of bio-piracy can happen, and those units can be taken out of community and then protected by entities such that the community can no longer grow them. And then on the other side, again, if you have been doing very specific work and have been taking plants and a population of plants and really breeding it forward to a specific purpose, a specific phenotype, how do you reference that work such that you can gain IP considerations for the work that has been done? So specifically for this project, in order to address those overall goals, we will voucher many plants within the community, so hundreds of plants within each community. These vouchered plants will be held as a special collection with the Candor Herbarium and will be tied back to the communities themselves, which will have vouchers of these plants. In each of the 6 legacy regions and in 2 urban regions, we will support community members in selecting which plants out of the assemblage of plants that are collected are of most important to the community itself, such that they can have additional genetic and phenotypic descriptions documented around those plants in the form of cultivar registration. It's a lot of prior art documentation will serve as a good template for moving those types of codifications through with other plants. And the work will also result in the documentation of community language around cannabis genetics. So what are the naming conventions that make sense to individuals in the community that have been working with these plants? What are the traits that are important to be describing when doing cultivar definitions, right? We have INUCP conventions for hemp. Okay, those aren't going to naturally translate over to cannabis. We have to look at many other things, sensory, olfactory ways of describing the plants, chemistries and growing conditions that are very different from our still cannabis, but sort of the hemp side of things.
Okay, so now into the last portion where I'll finally start talking some for y'all. Where part of this project is to also, of course, understand some of the genetic diversity both within and between communities in this plant in the state. So it seems helpful to first kind of talk about what we mean by genetic diversity. So ultimately, all genetic diversity originates from random genetic mutation. So a mutation happens, and then over time, more mutations can build up. And so how those mutations arise in individuals and those different alleles that come from those mutations get mixed together, that can happen heavily through just the process of recombination and sexual reproduction. And those are 2 important forces we'll talk more in a moment in particular in how it happens with cannabis. And ultimately, with genetic diversity, you also have forces that lead to the loss of that genetic diversity. So in this little cartoon, humans are awful and build a bunch of houses around these rhinos, and over time, we lose some of that genetic diversity. So we want to understand what forces could be leading to that. And what simultaneously, there are forces that could be out there that help maintain some of that genetic diversity. And it ultimately builds up to what sort of diversity we have in this species in the state. And another key concept to make sure that we're all on the same page of are these terms homozygosity and heterozygosity. And if we're talking about a single gene, all this means is whether or not you have two copies of the same allele in a diploid individual or two copies of different alleles. So that would be heterozygous. So sources and maintenance of genetic diversity in cannabis. Well, so first off, mutation. So there's been a lot of really great work done on looking at mutation buildup in cannabis over time. Certain lines are mainly clonally propagated. And so, right, if you have a mother plant growing over time, she can build up more of those mutations, and those can be passed along to clones taken off of that plant. And this particular project over here, they found that there are more unique variants that occur at the top of the mother plant over time. But in general, if you're keeping an annual plant alive for a really long time, you're going to build up a lot more of those mutations, and that will contribute to more genetic diversity being propagated. Another thing that I know y'all are familiar with is the mixed mating system that happens in cannabis. So, right, we have a plant that is primarily dioecious. So we have separate male and female plants, an XX female and XY male. And then there are certain cultivars that are monoecious, where they'll routinely produce both male and female flowers. And all of that does is it contributes to the sort of variation that we will find in cannabis over time. Of course, another way in which cannabis has a lot of diversity is a result of the fact that it is useful for many, many different things. We talked a lot about medicine today, but I know everyone knows it's used in many different sorts of products, textiles, even making plastics. It's an incredibly useful plant. And so those different sorts of selective pressures that people put on as they're trying to breed for their own purposes can also maintain different sorts of alleles in the species over time. And finally, there's this fun idea that we kind of talk about, and it's this what's referred to in sort of evolutionary biography or evolutionary biology as island biogeography. And so because of prohibition, cannabis kind of had it was sort of in these different islands in different populations isolated across the globe where people had these populations and there wasn't a lot of gene flow occurring among them. And that allowed for different variants to kind of evolve over time. Is that good?
Yeah, I think it's just a very interesting concept that there's a lot of negative things about prohibition, but the one, super fun, positive silver lining, if you can put it that way, is the amplification of unique variants because of this isolationist breeding practice that is very similar to the type of botanical and in the Galapagos finches are a really classic example. What this diagram is of, where you have a finch that is native in its land-based in its land-based area that moves to an island, and then as the birds separate onto the different islands, they become very uniquely adapted to the island on which they're at. And so the same thing has happened in cannabis, where you have different communities really stewarding these plants in a way where their goals for breeding and their areas where they were growing these plants really drove diversity in different directions. And so the result is really an explosion of diversity in a very small geographic space. So it's kind of fun.
definitely fun. And the point is that there are a lot of different forces that are influencing genetic diversity within cannabis. So here I kind of want to just go over certain ways in which we can look at genetic diversity and understand it. So first and foremost, there's a ton of genetic diversity in cannabis. This is well known at this point. So there are different ways to kind of try to measure that. So for example, this is just one study, Henry et al. From 2020 estimated that heterozygosity ranged between 0.03 to 0.5. And so that's a really big range for heterozygosity from different individuals, telling you that some are very variable. Remember that previous slide talking about homozygosity and heterozygosity. There are some that are more homozygous, and there are some that are very heterozygous. So that's a lot of variation there. Another plot that I bet many people have seen before are this plot right over here. This is a PCA, or principal component analysis. And the important thing to know when you look at a plot like this is that it broadly tries to take multi-dimensional data and put it into a two-dimensional space. And generally, the closer two dots or triangles or whatever shape on this graph, the closer they are, the more genetically similar they are for those markers. But it's important to note that just because two dots are next to each other, or even right on top of each other, does not mean that they are related, does not mean that they are genetically identical. And then finally, this plot on the bottom is called a structure plot. And this kind of gives us a sense of looking at population structure across different individuals. So each column in this sort of plot represents one individual. And it just shows how much of its genetic variation partitions to these different groups, which are these Ks on the right-hand side. So ultimately, there's a lot of different ways to look at genetic diversity. And we'll explore different ways to try to communicate this type of data from this project with people. But
Hey, I just wanted to jump in real quick, Nick. I don't know if you guys are trying to share a deck right now, but there's nothing on the screen share.
There's nothing on the screen.
Oh, I'm seeing it.
Me too.
Okay, then that's on my end. Sorry.
That's good, because the technical difficulties are always like, you never know if you're presenting because you're in this weird box. So
Yes.
okay, good. I'm glad
Okay,
that
now we're seeing
For
it.
saying something, I was like, oh,
Sorry.
look silly. Punchline is there's a tremendous amount of genetic diversity in cannabis. So why does genetic diversity in cannabis matter? Well, first and foremost, no one wants to live in a world with no diversity. And this is especially true when it comes to the plants we love. So I'm talking food and medicine. You know, what's good for the butterflies is good for us. We need diversity. And so that's just a blanket fundamental thing. Diversity makes plant populations resilient to environmental change or novel diseases. So having variation is a good thing for the longevity of using cannabis, and us in general and the globe. Genetic diversity is a toolbox for breeders and cultivators. So thinking about different alleles that different plants will have that your breeding stock you're not currently working with, well, that's how you can take that new genetic variant that you might really want within your cultivar that you're breeding and be able to intergress it. So things like disease resistance, flowering time, there's a lot of variation in that trait in this crop. Terpene and cannabinoid production, I mean, the list goes on. Variation is key in order for you to be creative and have this palette to work off of. And there are pros to heterozygosity, and the important point for that is this concept of hybrid vigor or heterosis. So essentially, the more heterozygous an individual it is, generally they'll be bigger than both of the parents and potentially yield more. So this is a routine thing done in a lot of crops. And that you can also potentially have some novel trait combinations and have the plant express herself in really, really exciting ways. But of course, I felt like it was important to also note that there could be downsides to heterozygosity, right? Say if I have a seed line and it's highly heterozygous, and I germinate a bunch of those seeds, they're going to be variable because of that. So there will be segregation issues and difficulty in plant uniformity. But in general, variation is a good thing, and we should all want it. Genetically characterizing cultivars the community cares about. So a part of this project, what we will be doing is we will be deep sequencing cultivars that communities from different regions think are worth documenting in such a thorough way. So this benefits those communities by generating a resource for them to use as they desire. But it also is really helpful as we try to better understand what a cultivar name means. So how things are similar versus how things are different. Do you want to do the dog example?
This is my classic dog example. Many of you who have heard me speak before, I'm kind of a crazy dog lady, but I think it's a very conceptually easy way to understand a very complicated topic when you talk about naming. So with a lot of the names that are in the community that are deeply rooted, like Bubba Kush or anything that's a sour, sour this, sour that. What does that mean? And how do we use the cultural language to help inform the genets or the taxons that actually are units that we want to talk about as cultivars? And so the way that I have always conceptualized this is with dogs. And so we know like maybe the sours are kind of like shepherds. So we know we have lots of different kinds of shepherds and they're all unique in their specific traits, but they're all in this larger category of shepherd. You have the same thing with sight hounds. There's many different flavors of sight hounds, and it's kind of like the Nebraska example as well, where you have these layerings that go more and more fine scale and fine tuned as you go from species to crop type, maybe broccoli, down to, Pennsylvania 62, a specific cultivar of broccoli. And I think the dog example is really easy for me because the categorical can be very structured in the things that keep it as a category. A shepherd has to do certain things. There's certain traits. A sight hound has certain physiological, their eyes are straightforward. They generally have a narrower skull. They have certain kinds of noses. There's characteristics that group all of them together so that then you can start to get the nuance, like what is the difference between an Afghan and a Saluki, where you can really parse it down. And I think if we take this framework into cannabis, it can be very helpful for us to start to categorize the bins where we can start to narrow down to the specific names that are either individual clone driven or very specific ecotype nuanced cultivar. That's my dog example.
Okay, so this part of the work will build off of the herbarium work that we're doing. And so we're going to we want to look at the impact of a licensure on genetic diversity of cannabis in California. So we'd like to do a comparative analysis of the genetic diversity between legacy farming regions with a different number of licensed farms and legacy farming regions that haven't had an opportunity or a path for licensure. So the study will provide a better understanding of the effects of California's legalization process and regulation on the legacy plant diversity and persistence, and how access to that licensure and the market dynamics affect this diversity. So ultimately, we're going to just compare different regions that have had this and see what does genetic diversity look like across those regions. Because sometimes the forces that are involved in modern ag limit genetic diversity, and of course, we want to prevent that. So figuring out what situations regulators are putting people under and how it's ultimately influencing this diversity in cannabis.
And so I think this really ties into the rhino example that was in a slide earlier. The impact of urban planning or legislation and how that affects the plants. You can't understand how to preserve genetic diversity if you don't know how the system is affecting or limiting that genetic diversity. And so it's really a proxy as to how to get an eye into what's going on with genetic diversity. As an ag state, one would think California has a deep vested interest in maintaining genetic diversity as a valuable resource for an ag-based economy that is so important to the state. And so if there are in fact things that are limiting genetic diversity and or reducing genetic diversity because of the regulatory framework, it might be really good for them to know that that's going on. But we'll see how the data fall.
So genetic data availability. So with community and individual permission, LeafWorks will digitize, select qualitative and quantitative data and make such data and further analysis available on the project website. Further analyses and interpretations connecting the findings of oral histories and other interviews with sequencing data will be disseminated in a final research manuscript from this project. And an important sample is chosen by the community to be deep sequenced to also provide a resource to the community to better understand their own plants. So in summary, cannabis is an amazing and diverse plant, and so is the community that has acted as stewards for this plant for generations. Voucher specimens will be collected and maintained in the Candor herbarium to act as a resource for that community, and genetic data will be produced for a subset of the key plants chosen by the community. And with that, we're happy to take any questions.
So I see one raised hand, AC Moon, good to see you. Did you have a question?
Sorry, I was having a problem with my audio. I apologize. Thank you guys so much.
We understand the technical difficulties.
Yes.
I was watching that, and I'm over in Humboldt working up in the hills, and tends to be like a little island off of its own. So thank you guys so much for what you guys have been doing. I've been watching you for years, and my intellect has a major, major crush on what you guys are doing. I've got the fattest crush on the information and your mission. Oh my gosh, I love it so much. So, but get to the question, because there's lots of people I'm sure that have questions, is okay, because I've been presenting to the ICNCP as well as ASTM about the nomenclature and the binomial nomenclature separation. And so I've been really trying to identify, as you said, how for me as a cultivator and somebody that's cultivated for 25 years, keeping the same genetics alive for almost 20 years in order to observe adaptation and mutation in several sectors and equatorial regions, I really have been trying to push the context that you guys are reaching, which is trying to make a hard decision on separating the different varietals of cannabis. I mean, cannabis sativa, if it was based on Linnaeus's structure of taxonomy or one of the other taxonomists, it really doesn't matter. It's just based on the fact that all of these things are traits or different variants of one specific plant. So when we look at the rose family and all of the Rosalis genetics, the way that it's written is very specific. And it seems, as a hustler that has transcended in a patented and working with patented genetics and worldwide stuff, I've been trying to figure out how to really lobby for ICNCP to really definably separate it. I found one scientist in France and in Europe that started speaking of cannabis spontanea. And so the cannabis spontanea is now in Europe being regarded as the ruderalis species or ones that may be affected by autoflower genetics. And so I felt like that was the beginning of being able to separate out that binomial nomenclature so that we can have epithets that really express the cultivars. Like this one's floppy, this one's tall, this one's got wide leaves, this one can handle tropics, this one can handle hemispherical changes, and trying to get those equatorial zones going. So as I reach these decisions, and I've been also collecting herbarium factors for about 20 years, I'm wondering if for you guys, do you feel like feminization or the use of like silver nitrate actually changes DNA sequencing? And does it affect it in a way that it should be
Boom, boom.
questioned on if it is an actual like solid parental genetics or if it has been feminized? Do you believe that that DNA changes and it inserts more hermaphroditic genes into the specific cultivar definition, since we're not using chemovar?
Yeah, I mean, it's a huge question, and it's a huge topic of research. I would say that the nice thing about the work we're doing is it kind of doesn't matter in some way, because we're at such a ground level that the decisions that we need help as an academic community interfacing with a cultivating community with the knowledge of the differences in these plants is really much more categorical and abstract right now. And what you're asking is so nuanced. And so in the beginning, I think the rose example is a very good one. So if you take the dog example and overlay a rose example, so like I said, shepherds have a certain job. So in roses, we have a very similar kind of approach where the first thing you
Absolutely.
ask about a rose is it a tea rose? Is it a composite rose, right? Is it 5-leaved? Or is it multi-faceted, right? Because those
Yeah.
are different groups. So now you can say, okay, I have a tea rose, but they're all, they're all roses. But
That point, though, is for us to indent
categorical.
those definitions into those Latin words that we can, as horticulturists or botanists, we can read those words, like you said, and it expresses that point. So I was trying to figure out if others agreed that it changes the DNA structure and in a way that may insert hermaphroditic genetics into the allele span. And
Yeah,
if it needs to be verified as we collect this data, should it be verified that this is a seed legacy? It's been in mother form for 20 years, or if this is something that is hermaphroditically reinvented through silver nitrate manipulation.
Yeah, well, you're definitely going to have different categories, much like the categories of like a tea rose or a climbing rose. There's these different categories. So you're going to have clonal plants, and then you're going to have seed plants. And within those seed plants, you're going to have normal seeds, that have your males and your females, and you're going to have feminized seeds. So feminization, from my perspective and from sort of a more general agricultural perspective, is really just a tool that's used to propel breeding. I think one of the issues is if you have a very diverse plant and you haven't done the work breeding it so that is more homogeneous, the act of feminizing it, how do I say this gracefully? It's kind of a waste of time.
Yes.
Still
All
an
right.
outcrossing event, because you're having the same, just because it's the same plant being used as the mother and the father, if it's really heterozygous, it's still an outcrossing event. So that's one thing. It's like it's a time and a place issue. Like feminization can be a very effective tool if used at the appropriate time in breeding, when you know what's going on with the heterozygosity of the plant itself in general. But you speak to another point, which is the propensity to herm, right? So we know that we have a mixed mating system. So we don't have a plant where all of the plants are monoecious or dioecious. We have a system in which we have both monoecious plants, the plants that have both male and female parts naturally occurring, not due to hermaphrodism and some kind of aberrant stress response. We have normal monoecious plants, and we also have dioecious plants. And in that dioecious system, we have most mother female plants, if you stress them out enough, whether it be by biotic stress, abiotic, whatever, stress, or SDS treatment, which is a different kind of stress, you can flipping the lights, whatever it is, like any
hormonal
stress,
response.
you can force them, or force is maybe the wrong word. You can incentivize them to freak out and produce pollen.
Mhm.
And so in doing that, we all should know that sex is not fixed, right? If you have a female that can also produce male gametes, we're on this sliding scale of sexual evolution, which happens all the time in plants. Sex is a recurrently evolved trait in plants. And sex chromosomes are super interesting in the plants. Like sex is determined in very different ways in different groups. And so we're really just scratching the surface on how sex is truly determined in cannabis and how variable that sex determination is across the entirety of the species. So there's some really good papers. There's a gentleman, Dr. Alex Harkis, who works at the Alpha Hudson Institute in Alabama, who is, I venture to say, the world's expert on sex chromosomes in this particular arena, and is doing an immense amount of work on the evolution of sex in cannabis. And so his research is going to be very enlightening in the coming years to answer some of these questions. It's a big topic, and I do think it's important and I think what it really nails is—
Thank you. I really appreciate it.
Yeah.
Right, there's seeds, there's clones, there's a lot of gray in between.
I also saw a talk recently about SDS, and I just want to encourage everyone to use proper PPE when you use that, because that stuff is pretty gnarly.
Yes.
And I really was just digging in, and since you guys both work in genetics, I think my main core is, do you believe that the use of those silver nitrates changes the DNA structure? Because I feel that DNA altering is—
It seems like it does act as a mutagen in addition to changing expression.
All right. Okay,
But,
great.
but
there's
there
lots
needs to
of-
be
there's-
more
there's
work
lots of
on
mutagens. UV is a mutagen. So I think the next hand up was Taslim Tagore. I hope I pronounced it.
Thank you for your time.
And I just
Thank you
wanted
very
to—
much.
Yes.
Can I just jump in real quick and just encourage folks to— we've got the Q&A box. So if folks want to also be putting questions in writing, then we can kind of tag team and get more engagement. So just want to remind folks that's there. Happy to pass it back to you, Tasmin.
Okay, thank you. So I'm kind of new to the conversation. I definitely understood very little of what you guys just discussed with our last question. My question is really just surrounding the medicinal healing properties of the plant versus it feels like so much of the conversation has shifted to the recreational use. And I don't even know if there are folks who are preserving strains that are, that are intensely medicinal, that are teaching, that are helping, the conversation to enlarge beyond recreational use and consumption and trying to get the numbers up in the state, da 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 da. And I guess I just personally don't want that part of the conversation to be lost, because that's how we develop this intense respect for the plant and develop, a relationship that's sustainable and deep and mutual. And I just don't know where to look to connect and learn and feel part of a larger community that cares about these same issues.
Well, you just spoke to our hearts so genuinely. We're medicinal plant people. That's the plants both Nick and I have studied, and a large portion of our team have been medicinal plants. And I see a hand up, which we will speak to in just a moment. Wade Laughter. There's people on this call who are doing exactly the work that you are questioning. The medicinal community within cannabis is deep and strong. Licensure has not necessarily been friendly to a lot of those organizations. And so a lot of this understanding of diversity and how diversity is atrophying at the peril and toll of licensure and a rec system that doesn't necessarily add enough value to those medicinal plants for lack of research. Because once we figure out something's really good at cancer prognosis, humans care about themselves, unfortunately, more than anything. And so I do feel like we will have a shift, but we need more time on task to get there. The medicinal community is deep and wide, and so I would love to connect with you offline to connect you to some individuals that could maybe give you some more community within that area, because there are many individuals working on that. And that is part of the most important, in at least my worldview, aspect of diversity maintenance and understanding, is that just because something looks good in a bag in a jar, like, that's important. I'm not poo-pooing that. That is a very important thing, but it is not the only important thing about the preservation of genetics. And so we are interested in that. We can't wait to help in any way we can with not only documentation through the herbarium, but just connecting and being part of this community, and the medicinal side of things is fundamentally important.
Okay, great. I'll reach out to you guys and we'll follow up. Thank you.
Love it. Thank you.
Much.
And I think Oliver was the next with his hand up, so I would love to
Hey, Oliver.
hear you.
Hi, guys. So good to see you. You'd see my face if the internet wasn't so bad. I'm terrified to turn the video on. But pleased with your presentation, and really appreciate all this work. So in Big Sur, on our epic journey to the glorious goal of a cannabis appellation someday, we have been met with regulation that didn't work for our community. We rejected it. And we are under a gray area of prohibition. So my question is going to kind of stem from that. The unlicensed legacy crowd that is very interested in participating in this and see the work as so important. But the prohibition of our communities has led to a reintroduction to war on drugs 2.0 in a way, and then politics is wild, just all the politics, all these conspiracies, what have you. Number one question I get is about the protection of this study. And really, these wild mountain men believe that the state of California is against them and that they are in cahoots with the businesses that dominate the space. And knowing that it is a DCC sponsored grant, we have great faith in this system. But as I am explaining this to people, we do find it challenging, and especially the ones that have been here the longest, right? That really are the joys, the nuggets that we need in this study. Would love just a little bit more on that and how you guys are feeling. And I know there's been a lot to that, but yeah, that's my question. How do we separate church and state a little bit so that our lovely legacy Californians are more enticed maybe to participate?
Okay, I can definitely take that. That is something that is not new to us. Interacting in this space it's always been that dynamic of fear, and a lot of the academic institutions that have interfaced with the community have done nothing to help that. And so we are really here to be very transparent about what the study is and what it's not. And so I think it's helpful to be able to talk so plainly and so interactively about genetics, because there are certain things genetics can do, and there are certain things genetics can't do. And I think those lines are often blurred, and that's what creates the fear. And so with the vouchering portion of the project, it is the safest way to document your material, because nothing can be propagated from that voucher. It is an inert physical representation through the pressing, and it is an immutable object that is static in its timing when it was taken, so that you can associate a community or a group with that particular plant, but nothing can really be done with that plant to propagate it, right? You can't steal that plant through a voucher. The only thing you can do is point back, this voucher existed at this time, in this place, with this community or individual. And so it gives power and litness to those groups to say, no, I have ownership of this. Another thing that's really important to remember is no sequencing will be done on anything that isn't expressly put forward as something that will be sequenced in this project. There's a very finite budget, and monies will be used specifically in the ways that are directed by the community. So if we have issues around, like, how do we define this cultivar? We're going to sequence these 5 things that all have the same name and figure, there's different ways that can pan out, but that will all be directed by the communities themselves. And then I think there's also a little bit of a reality check that always has to happen with genetics, in the sense that I can go to the grocery store and buy 20 different apples, and I can sequence them. Those things are actually living. I can tissue culture them. It does not mean I own them, right? And so you have a situation where you can't produce a living plant out of the material we're taking. It's very specifically designed to be desiccated, pressed, it's inert. And the genetic sequence itself, there's nothing sacred about that. And I think that's something that geneticists have not done anyone any favors with, because there's like, woo is the wrong word, but this sentiment around genetics that it's like the ultimate key to do everything. And it's just another tool in the toolbox. You have to actually have that plant in hand to do anything with it. And if you have sold that plant into the market, anyone can sequence it. There are no rules about sequencing in general. And so the power of the sequence actually comes from the association with the voucher and the placement of the plant in the hands of the community. So I don't know if that helps.
That helps a big time, actually.
But it's
that's a big part of it, because we're figuring out our own legal protections, and that misuse is the number one thing that comes up. Are they going to steal my plants? What if my plants are out there already?
And then my answer to that, Oliver, is always, this is the quickest, easiest, least financially impactful way to protect yourself. Because if I, big corporate, whatever, come in and say, this plant's awesome, I'm going to take it and call it something else and get some plant protection on it. All of a sudden, you have a very tangible saying, A lot of the utility patents are the same way, where you have organizations saying, we're patenting plants that are between 1 and 6% THCV and under 5% THC and over 0.1% CBD. Okay, how many plants fit into that? Many, right? So the idea that you can get a utility patent on that is insane. And the reason it happens is because there are no resources to say there's nothing uniquely genetic. There's no unique genetic that is only that thing. So really what this process is creating an arsenal or a repository of information that can be utilized by the community to do exactly what you're saying, to protect themselves against thievery. So, and I love to talk about this and dive in, because part of this really is what is the most useful to the community, because that's what we want to do. I'm going to move to Wade. I'm so excited to have Wade on the call. I know that he typed something in, but his hand is up, so I would love to hear his voice if that's possible. But I can also read the question if you need me to.
Hello, Eleanor. This is Wade. And yeah, I typed what I could of my question into the Q&A, but I'll just give you a quick, bring you up to date on what I've been doing lately. I've, since 2011 or so, been very focused on pediatric epilepsy, and specifically looking for versions of the cannabis plant that provides chemistry that stops these kids from seizing. And I'm talking kids who seize 100 or more times every day, day and night. It kills some of these kids. And yet, if we're lucky and persistent, we can find versions of the cannabis plant that literally stop these seizures in their tracks. No more seizures. No more seizures. And my issue is, how do we preserve those genetics? Because what's true is the genetics that are just right for this kid over here that has an identical diagnosis, it
No.
might make his or her seizures worse.
Mhm.
And so there's clearly a very specific chemistry going on that provides this therapeutic benefit to these kids. And the diagnosis is not any guide. And as a general thing, I'll say that, like at one point I thought, oh, well, all these kids need is really high CBD plants. Well, that turned out to be wrong. I remember at one point, trying to sort out what was happening for one of the kids I was working with. I had 6 different high CBD plants, not much THC at all in any of them. And one of them gave really dramatic relief from his seizures. But one of them, if I was just looking at THC and CBD, they were the same. But this other plant actually made his seizures worse. So I'd really love to have you wave a magic wand and tell me that you've figured out a way for us to identify the chemistry of these plants and how to preserve that chemistry for the community. And specifically because so few of these kids are around, that there's no money to be made in it.
Yep.
It's really a matter of compassion. And I got to say, the cannabis industry, that compassion flower has wilted quite a bit in the heat and dryness of our current cannabis industry here in California. I don't know anybody who's got a smile on their face that's obeying the rules. So anyway, here I am. I'm a person with no license or permit, but I'm plugging away at trying to find the plants. And I have no interest in protecting what I find. I want these kids and the community to have access to these plants. So I'll shush and let you tell me you found a magic wand, Eleanor, or that Karen has found one.
Oh man, well, we're looking for those types of magical wands. And Wade, it is always so lovely to hear you speak and the devotion that you've had not only to the plant, but the community that's really used these plants in a medicinal fashion. You're truly an inspiration, and you've been one of our cheerleaders since the very beginning, since before the herbarium was even founded. You were exceptionally devoted to the work and saw the vision of the work. And so one of the things that I was speaking about earlier was documentation. So one of the things an herbarium is exceptionally good at is documenting things. So in your example, one thing that would be lovely is to press all of those 5 plants. And then you, as a user of the herbarium, you can start to say, okay, these are the plants that always work for this child. This is the plant that always works for this family. And then you can start to utilize that resource of the herbarium to say, okay, how can we compare these things? To then use it really as a tool to figure out what is it about these plants that's driving positive prognosis? What holds these plants in common? So it's the same kind of question of a farmer, I want to grow on a new property, but it's a medical question, which is, I have a particular issue. I know that there's potential for positive results using cannabis, but those results are variable. And this is another reason why names are so important, because if I have epilepsy and Nick has epilepsy, and we're in a clinical trial or some kind of clinical trial so formal, some sort of group where we're monitoring through any kind of healthcare professional how our prognosis is, if we're sitting in different homes taking a tincture and we're not sure we're using the same plant and we have different responses, it becomes academically impossible to disentangle. Is the positive prognosis or the positive results I'm having using this because of my chemistry responding, which is exactly what you're saying. These kids have different responses to the same plant. So that naming convention is even more important in medicine, so that we can start to talk about the variation in the humans that also affect the effect of the plant. And so it's layers of importance. And then you can start to assimilate data using the herbarium as a resource. So you can voucher things and submit them. And then you can say, I need these samples for this analysis. right? And so that's one of the more long-term, very important areas where the herbarium can be exceptionally useful. Making sure we're holding the plants constant so we can know if they work and we can start to understand human variation in their endocannabinoid responses to these plants. And then also understanding, okay, it's this kind of plant. So maybe I need this exact cultivar, or maybe these 3 cultivars are similar enough in the chemicals they produce that either one of these is interchangeable for my child or my human that I need this medicine for, such that I can have more options of medicine. Because part of the problem is also if you have a medicine that is working very well and then all of a sudden no one is growing that. How do you fill that gap with the farming too? And so really the idea is to be able to use the herbarium on both sides. And if we find that magic wand, we're going to use it, and we're looking, but really what we're doing is what we can to help provide a resource by which these questions can be answered in such a way where we get real information.
Thank you.
Much love to you, Wade. Thank you so much
you.
for being here.
Oh, no, thank you for doing this.
So there's one question in the Q&A from Jasmine Cardenas. I'm a steward of this amazing plant and came to the first panel discussion representing Network of Applied Pharmacognosy. I work in the industry as well as in tissue culture sector of the tissue culture sector of cultivation at Finest. I'm so excited to witness this history in motion and all about genetics preservation. Would tissue culture be an option for preserving these genetics by way of cryopreservation? And how can I be a part of this?
Yeah, you want to?
Well, I would say like we're certainly interested in thinking about how tissue culture, because that's an important way in which this plant is propagated as well. I know that the herbarium, just by the way that it operates, is kind of the main way we want to preserve specimens. They last hundreds of years. There are herbarium vouchers from the 1600s still, as long as you keep them in the right condition. It doesn't require, a cryo freezer. But I certainly know that we are open to trying to figure out ways in which how to integrate that important facet of the industry into this project.
Yeah, and I think it's really nice as a tool for individuals that are cultivating to be able to use tissue culture to preserve plants where, nursery space and keeping mothers, that's a huge amount of work and time and money. Not to say tissue culture is also a financial commitment, but it is a very amazing tool for individuals to be able to preserve things long term. And, we need all the tools we can get.
Thank you for that. Sam, how's it going?
Hey guys, I just wanted to pop in here if there was no other questions. I didn't want to pop my hand up before others. So are there any other questions in the Q&A? I can't see them all.
I think right now, at least in the Q&A, that they've been answered. But please,
yes, I believe so.
Raise your hand.
Thank you for all the kind words, everyone.
Yeah.
Okay, I'll try to be brief here, but I just basically wanted to consider. So I met up with, I'm here on the Big Island seeing just amazing cultivars and plants that have been stewarded here for generations as well. Very similar to California. I always call this kind of a legacy producing land out here. But I met with a friend of mine who grew up in Willits, California, up Sherwood Ranch, generations of cultivators. And she had just recently thrown away a stockpile of genetics because she was like, I don't see any future in the industry. There was nowhere that I could see that these would, I know it makes me so emotional. And she's a longtime family friend, and it would just killed me to know. So I'm just kind of curious, like, what message can we send to the growers, the breeders, the people, whether they have entered the regulated space? And, the study does not restrict us from, participating with anybody who's a home grower, a hobby grower, a breeder, a seed saver. So I'm just curious about how we can kind of convey the messaging to folks that whether they choose to participate and get involved in the study or not, just that the tools that and the research and the data and this amazing project and what it can bring to our communities and how potentially it can, steward the future of some of these breeders and, and some of their unique cultivars, many of which, as Wade mentioned, the passion for medical was the pioneer at that time. Like, these were the folks that were breeding for cancer patients and for, epilepsy and for glaucoma and these different things. So I'm just really passionate about that side of things and how we can kind of convey to people, like, don't throw away all of your stuff. Like, it is not, just because the industry is in shambles in California does not mean that, like, there's no future in this biodiversity. And the second fold to that question is basically, like, where have we gotten to with this, what we call pollen chucking and gelato inbred genetics, that, people are seeing these, nodes pop out of places they shouldn't pop out of. And I like to say that's not some magic mutation. That is, just like with the hop latent virus, like, these are things that you're going to see when you inbreed and when you get further away from that genetic biodiversity, right? So that's my kind of twofold question. I know it's a lot for the last few minutes, but
it's great. I would first like to make a plug for a colleague. So Zach Stansel is on the call. He works at the USDA. He's the curator of the hemp seed collection. If you're in a position where you just want to throw stuff away, I would suggest hooking up with him and considering to donate your seeds to the USDA. It's an exceptional way, if that's really where you're at. Like, I don't want this, I don't see a future, to give those seeds to an organization that will steward them and make them available to other cultivators to use in their breeding programs. And so it's a way of preserving that biodiversity. And I know right now it is a hemp seed collection, but I know that cannabis is a growing, important agricultural crop. And so that's one avenue. And I don't know if, Zach, I'm putting you on the spot. You don't have to say anything if you want, but I would love for you just to introduce yourself, because you're a phenomenal resource to this community.
Yeah. Hey, well, first and foremost, thanks for this wonderful event. So cool and interesting. And I love the work that y'all are doing. So yeah, my name is Zach Stansel. I'm in Geneva, New York. My job is a germplasm curator, and I work for the National Plant Germplasm System. So at our lab, we keep the world's largest apple collection. So if anybody's ever read like Botany of Desire, for example, that was, a chapter of his to come out and talk about kind of the similar work that we do. So I was very lucky to be hired to start the hemp germplasm collection up here. And we don't buy seeds, we don't sell seeds. We're a resource to the broader scientific and research community, as well as we serve people with educational objectives as well. So broadly speaking, our goal has been to collect diverse germplasm.
It's like a bunch of different offices. It used to be like a
Sorry. So we collect and conserve germplasm from all around the world. People have donated different land races and varieties and old grain and fiber types from Europe, that kind of stuff. And we work with different folks to characterize it phenotypically and genotypically, and then we make all of that data publicly available. So that's kind of the big part of our goal is I'm basically a seed librarian. So if that's of interest to you, I'm happy to share my email address, and I'm always happy to talk about this kind of stuff. But yeah, thank y'all so much for this wonderful event and your wonderful work.
Thank you.
Thank you.
And so it brings up seed libraries, and these are things that, I have a long history in herbalism and medicinal plants, and seed libraries are part of that. I think we see that in California right now with the fires and the Altadena seed library, just how impactful having local seed libraries really are. At Candor, it's a little bit too close it's too close for us to be a seed library. It would muddy the waters of our objectives, which is really to be a resource for herbaria vouchers that can be referenced, and we very much want to stay in our lane. But being involved in helping with seed collections and seed libraries. There's something that we would love to work and volunteer our time for, but it's not work that we feel the herbarium can do in this moment of time, given the political, mental state that the industry is in. It's just too close. And we want to be very mindful of our boundaries and be respectful of the community. But that is definitely projects that we are wholeheartedly behind and would support both financially and with our time. So seed libraries, let's go. Maybe that's version 2 of what this group is going to accomplish. In any way we can push that ball forward, we're happy to help. Yeah. And then the second part of your question was really about jog my memory, please, Sam. Did I answer your question fully enough? Okay, Scott, your hand is up.
Absolutely. Thank you.
Yeah, guys. I helped to curate a small seed bank at Cal State East Bay in the Anthropology Museum. So if there's any way we could be involved, let me know.
Awesome. Well, I would love to connect and just chat with you offline. Seed banks, and especially herbal plant, medicinal plant seed banks, are a personal passion.
Absolutely.
So
I'll try to find your email.
Well, I'm happy to jump in if you guys are ready to wrap. And just want to thank everyone. Amazing conversation. Thank you so much, Nick and Eleanor and everyone. I wanted to flag that we have the last in our series of webinars coming up in 2 weeks. So on January 31st, I'm going to be presenting with our team around the research track on intellectual property and public policy. So that's going to be January 31st at noon, and we'll have registration open this weekend up on our website, and look forward to seeing folks there and having that discussion as well. So thank you guys all for everything that you all are doing for the community and for the plant and for each other, and have a wonderful weekend.
yeah, thank you so much for joining us. This is wonderful to get to talk about this.